section, we will try to understand how energy is produced, and we will also learn many terms that relate to the topic of metabolism. There are various reactions in catabolic pathways, which means that breakdown of larger molecule, and these pathways will generate energy through production of ATP molecule. What happens is that a molecule such as glucose, which the formula of glucose is C6H12O6, a molecule such as glucose. So here is the molecule of glucose that I wrote down. These molecules of glucose have potential energy stored in the bonds that they have. In other words, when we have the carbon-hydrogen bonds, carbon-oxygen bonds, and so forth. So if I was to, this is not really the true structure of glucose. This is, I'm trying to get the razor. Let's see, where is my razor? So in other words, in any chemical molecule, there are elements that are holding the two atoms, and between these atoms there is a high energy bond. Usually the high energy bond is associated with the carbon molecules. So when we break these carbon molecules, that energy that is released is then going to be trapped over a certain number of series of reactions. So let's go on to the first terms up here. In order for us to understand completely all the terminology, it's important we understand what is oxidation. Oxidation is called as the removal of electrons. It's the removal of electrons are going to be emphasized by the letter E, which is usually represented by electrons. So you will see electrons being removed from an atom or molecule, and then the reaction will proceed. A molecule that will remove an electron is said to be oxidized. So this molecule over here is going to be oxidized, which is the one in yellow. The molecule that accepts that electron is the one that is called as reduced. So in this reaction, notice the molecule B over here is actually accepting the electron and becoming reduced. The molecule A has lost the electron and is now oxidized. And these reactions are also called as redox reactions. Redox reactions are those reactions which are paired reactions. So for example, if I was to write down a molecule A converting to a molecule B over here, and this molecule is releasing electrons, and these electrons then convert. I know this is kind of difficult to write with this pen over here today, is moving from C to D. So notice that there's a pair of reaction that is going on between two sides, and these are called as redox reactions. So that there are paired reaction. Whenever you have oxidized, it's always going to be coupled with another reaction, which is a reduced reaction. In biological systems, the electrons that are associated with losing are always followed by the removal of the hydrogen atoms. So notice up here we are showing that electrons are being removed, but that electron is followed by associated with the hydrogen atoms. And because most of these reactions will involve the loss of hydrogen atoms, so these reactions are called as dehydrogenation, which means that it's reactions in which hydrogens are being lost. Many times we will represent these reactions like with -2H, which is representing that there are two hydrogen atoms being lost. Now let's look at the metabolic pathway that is shown up here. In this metabolic pathway, glucose is shown as a substrate. Glucose is a molecule that has, which I was trying to give you the formula up here, C6H12O6. So that's the formula for glucose molecule. which means that it has many bonds of carbon, hydrogen, and oxygen in them. If that is the substrate molecules, that's the molecule that has energy stored in it. The energy that is stored is actually a potential energy, which is the chemical energy that's stored in these bonds. When the fate of in carbohydrate catabolism, which means breakdown, is broken into three major steps. The first step is called as glycolysis. The second step is the Krebs cycle or the tricarboxylic cycle, and the third step is the electron transport. Now, many of you have already done this these topics in other biology classes, so this should be a good review of what you've already understood. So it's a good refresher. The first stage is glycolysis. Glycolysis occurs in the cytoplasm of the cell. In glycolysis, the 6-carbon molecule of glucose is broken down into 2-3 carbon molecules. So at the end we will have 2 molecules that are going to have 3 carbons each. It does make sense because we started off with a 6-carbon molecule. Now what it requires is a startup of energy. Remember, sometimes, and then you also release energy, so I should actually have energy shown over here. That ATP is required to initiate this reaction. So you need 2 ATP molecules in particular, more like a primer. Just remember how you do your lawns, and you have to provide a little bit of energy as a primer. So this ATP that initiates this reaction, which can go forward, the reaction of glucose converting to pyruvic acid is a 9-step reaction, and at the end of this reaction, glucose molecules are split into 2 pyruvic acids, 2 ATPs are released, and 2 NADH molecules are taking the hydrogens away. Now, if it starts with 2 ATP, it actually makes 4 ATP. The net gain that is shown to you is 2 ATP. The pyruvic acid, in the presence of oxygen, is taken up by the mitochondria only in eukaryotic cells. In prokaryotes, this cycle is not going to take place. So in your bacterial cells, there will not be no tricarboxylic acid. However, in eukaryotic cell, it does take place. So we will understand the entire metabolism up here. Now, in this reaction, which is tricarboxylic acid, this will result at the end with a cyclic form of many series of reactions. At the end of these reactions, what's happening is 6 carbon dioxide molecules are released, 2 ATP, 2 FADH2, and 8 NADH molecules. This is a reaction which is actually a cyclic reaction. It's a reaction in which pyruvic acid actually enters the tricarboxylic acid in the form of acetyl coenzyme A, and acetyl coenzyme A will enter this chain of events. Okay, let's go on with it. I was trying to pause just to see where the slides were, and we will do this in a lot more depth later. Now, at the end of tricarboxylic acid cycle, many electrons are being released, and these electrons are then taken up by the third set of series, which is called as the electron transport chain, which occurs in the membranes of prokaryotes and in the mitochondrial membranes of the eukaryotes. And in this series of reaction, electrons are being shuffled through a bucket brigade, and eventually at the end of this reaction, ATP is formed, and the oxygen is removed. Actually, in the presence of oxygen, electrons are being accepted by oxygen, and it forms water. So, and the summary that you have seen on this chart is actually summarizing all the reactions that we will do later on, which I will do a little bit more depth. Now, Glycolysis will yield 2 ATP molecules. If oxygen is present, so in the presence of oxygen, the process is called as aerobic respiration, and that will yield 36 ATP. Actually, it yields 34 ATPs. I'm going to write down 34 here because 36 is the total. And if there is no oxygen present, then there is no more ATPs that are formed by this reaction. So what is ATP? ATP stands for the term adenosine triphosphate. Adenosine triphosphate is made by phosphorylating ADP, which is a diphosphate with an inorganic phosphate. So a diphosphate that has two phosphate bonds. Pretty much it's a molecule. If I was to draw the adenosine here, and then you would have phosphate bonds, another phosphate bond. So these are the two high energy bonds, and another inorganic phosphate can be added to then give you a third phosphate bonds. That's shown over here. So you've got, notice over here, you've got the high energy bonds. In glycolysis, which is the first step in this process, it will make ATP. The A is missing over here from glucose. It converts glucose to pyruvic acid, which is a 3-carbon compound. So here is a 6-carbon compound. Here is the 3-carbon compound. And since there will be 2 of these because there's one 6-carbon molecule. Some bacteria can also break down other molecules, and those alternative pathways are called as the pentose phosphate pathways. And in these molecules, we have a 5-carbon sugar instead of a 6-carbon sugar. And in such bacteria that allow to do that are the E. coli and Bacillus subtilis. Now, once oxygen is present, the fate of glycolysis is going to be within this part, which is the Krebs cycle. If oxygen is not present, then what happens is the pathway actually enters the pathway which is called over here, which is the fermentation pathway. So this is lack of oxygen, and this is presence of oxygen. So in the presence of oxygen, you will have one pathway, and notice up here you've got a little dark division up here, so it divides the pathway in 2 different ways. Now, before it goes on to this pathway, there is a preparatory stage, and the preparatory stage is the one in which the glucose is going to be converted into, the pyruvic acid is going to be converted into acetyl-CoA. So let's look at a little bit more depth of all the stages in glycolysis. Here is shown to you 6 balls or 6 blue balls, which represent 6 carbon atom. Notice ATP is being added as a primer. As I said earlier, that you will start the reaction with molecules of ATP, and then over the process you get 2 3-carbon molecules. Each and the total would be 4 ATPs that are formed. So you have step 1 in which glucose converts to glucose 6-phosphate, then glucose 6-phosphate converts to fructose 6-phosphate. These two are isomeric forms, so in a reaction in which the placement of the phosphate group of the carbon group has changes, and then a second phosphate is added. That's why you have a diphosphate, and then the diphosphate molecule splits into the 2. Now, each diphosphate molecule follows a series of more stages to end in the molecule of pyruvic acid, and here you can see 2 more ATP are formed. So if you go, let me go back on the previous reaction. We have 2 ATPs forming here, and we have 2 ATPs forming here. But since the reaction to begin with requires 2 ATP as primers, we call the net gain as 2 ATP, and that would make sense to you now. Notice along with that we have NADH that's given on this stage, and another molecule of NADH which is actually going to be shown later on. So, and those would be the NADHs that are formed. Actually, no, there are two of them because remember there's one NADH coming from one 3-carbon molecule, and the other 3-carbon molecule is going to release another of these steps. Okay, so now once pyruvic acid is formed, pyruvic acid does not enter the Krebs cycle or the tricarboxylic acid right away. The pyruvic acid has to convert into a molecule which is called as acetyl-CoA. Acetyl-CoA is a 2-carbon molecule, and this is a 3-carbon molecule. So what has happened over this stage is a molecule of carbon dioxide has been released as 1 carbon, and we call this process as decarboxylation, which is removal of the carbon dioxide molecule. Now, the pyruvic acid, the acetyl-CoA, that is going to come and enter a next stage if oxygen is present. So acetyl-CoA, which is coming, which is the molecule that has 2 carbons, will combine with another molecule that has 4 carbons to form a citric acid molecule that has 6 carbons. The citric acid molecule then goes through this shunt or this ring of intermediate steps in which NADHs are released. So notice we have 3 stages of NADHs being released. We have 2 stages of carbon dioxide additionally being released, or is it 1 carbon dioxide? And 1 carbon dioxide was released in the process that just took earlier. So that's those are the stages of carbon dioxide. And we have FADH that's being released, and 1 stage in which ATP is directly formed. So a lot of activities is going on through the Krebs cycle. It's really important that you remember as to what are the details going on in here. Excuse me. So these intermediates that are present in Krebs cycles are all written in this chart, and many drugs are synthesized to break down these intermediate molecules. So some drugs are metabolized similar to these. For example, citric acid, isocitrate that has 6 carbon, alpha keto, 5 carbon, succinic, fumaric, malic, and oxalic. They all have 4 carbon compounds. So one way to combat a bacterial infection would be to have drugs that can break down these molecules. Now, eventually, the NADHs that have been released from Krebs cycle and from glycolysis are all taken by the carrier molecules, and the carrier molecules were the NADH. Those were the carrier molecules. The NADHs and the FAD bring these electrons through a chain of events, which are the electron transport carrier proteins. So what happens is, notice up here, the NADHs are throwing the electrons that pass through this carrier cytochrome, which is and it pushes the electrons from the inside to the outside of the membrane. Now, these electrons are followed by hydrogen ions. And remember, earlier I said oxidation is when you remove electrons, and these electrons are followed by hydrogen ions. So eventually, as more electrons are being pumped through these membranes, there becomes a gradient of hydrogen ions, which means that there are more hydrogen ions pumped across the membranes. So when there are more of these hydrogen ions, these electrons, they keep shuffling them through these electron carrier proteins, more like a bucket brigade. Eventually, the electrons are then pulled back into the inside of the membrane through a unit which is shown over here, and this unit is very unique. And this is when it comes back through this unit. This is actually an enzyme or a protein, a molecule that's called ATP synthase. It's more like a churning machine. When electrons are being shuffled back through here, then these electrons are followed by the hydrogen ion, and they are eventually accepted by oxygen to form water. On the other hand, this energy that's coming back is helping to form ATP molecules. Since if you go back up here, you notice how many NADHs we have. So the more electron carriers that the aerobic reactions can produce, then these steps, these will all be shunting to these electron transport a chain, and eventually give rise to a lot more ATP bonds. I'm going to show you an animation on these in class, so we will go over it again. So at the end of this process, what happens is the glucose, which is the molecule that has potential energy stored in the bond, is broken down through dehydration or hydration reaction. That is, water molecule is used, and ADP and inorganic phosphates help to form the ATP, and glucose is then broken down into carbon dioxide and water. So this concludes our anaerobic step. I think I'm going to take a break here, and then we will go on. Well, actually, we could go on. We don't have a whole lot. It's only 21 minutes. You can take a pause if you wanted. It won't get that long of a lecture. It won't get that big. Now the fate. I'm going to go back just to show you as to this reaction up here. Remember, we said if the oxygen is present, then this part of the reaction goes on. In the absence of oxygen, what happens is fermentation, which is what we are going to learn now, which is the second step, in which we will see, which is also called as the anaerobic respiration, or the respiration that does not that takes place with the lack of oxygen. Now, when final electron acceptor is not oxygen, that is absence of oxygen, various amounts of ATP is produced. Actually, it's much less, and it's a slower reactions. It uses some parts of the Krebs cycle, but very ineffective. So here we can look in this diagram over here. Glucose is shown to us to break down into pyruvic acid. This pyruvic acid, instead of going to the Krebs cycle, now will form two other molecules. One is the ethyl alcohol, and one is lactic acid. Now, which one it forms depends on the source of the cell. For example, in muscle cells, it will form lactic acid. In yeast or bacterial cell, it will form ethanol. Now, in this shorter pathway, carbon dioxide is released, and you have a 2-carbon molecule. Very little energy is formed any further. So this process, which is the anaerobic process, is also called as fermentation. So fermentation is what causes yeast to release carbon dioxide, which actually gets the bread, which will make our flour rise or bread rise, and you get formation of ethyl alcohol, which gives that tarty taste to bread that has yeast in them. So here is pyruvate, as you can see in the center, and here are many pathways on the side that are going on. Pyruvate then can be converted to many end products depending on which particular organism. For example, in Clostridium, it will form butyric acid. If it's propionic bacteria, it will propionic enter. In enterobacteria, it's going to form 2,3-butanediol. In Proteus, it will form carbon dioxide and water. So many pathways are formed. If bacteria only produces lactic acid, this is called as the homolactic bacteria. No additional ATP is formed in this process, and that's the most important thing, is like we don't form any more ATP. In alcohol fermentation, which I pretty much told you earlier, it's the bacteria that produce ethanol and carbon dioxide. It also happens in certain, for example, yeast cells. Bacteria that will produce lactic acid and other alcohols are called as heterolactic bacteria. So here is a summary of all the reactions between carbohydrates, lipids, and metabolism and proteins. You can see here is your main pathway, which is the starch and the breakdown of the carbohydrate, mainly glucose, into these three pathways. Now notice enzymes, which are proteins, and lipids can also enter the main pathway through either the acetyl-CoA formation, or later in the Krebs cycle, or in the TCA cycle. So now this is a little summary from your book. In anaerobic reaction, the electron acceptors are not oxygen. Oxygen is the final acceptor only in aerobic reactions. In anaerobic reactions, it could be nitrates. You can have sulfates, or you can have carbonates. And these would be the different molecules that will accept those electrons to either produce nitric oxide, nitrogen and water, hydrogen sulfide and water, and methane and water. So methane, as we know that many bacteria will produce methane, some will produce sulfur gas, and this is the nitrous oxide. And the reason I'm giving you these is because I know it's flipping. I don't know what's happening with my pen today. It's acting a little funny today. These gases can be produced by bacterial contamination or bacterial growth. So sometimes you have this foul smell, and it's because of any of these gases. Here is a pathway in eukaryote and a prokaryote comparison. Notice in glycolysis, both of them are taking place in the cytoplasm. The intermediate step takes place in the cytoplasm. The Krebs cycle can only take place in the mitochondrial matrix. In the prokaryotes, some of them, it will take place in the cytoplasm if they have those intermediate molecules. And the electron transport chain in the inner membrane in the eukaryotes and in the plasma membrane will act as the surface for electron transport chain. Here we can see the comparison of ATP produced at each cycle. Notice that a glycolysis only gives you 2 versus Krebs cycle, which gives you a lot more. And here is the actually, and this is the one in the complete oxidation of 1-glucose, and here is actually the ATP production of by 1-glucose molecule. So notice up here we have a total of 30 ATPs from NADH and 4 from substrate level phosphorylation. I really didn't give you a difference between substrate level and oxidative. I can do that over here. Oxidative phosphorylation is to do with electron transport chain. If you go back in the cycle, which I can go back in a minute, I'll show you there are a few places that ATP is formed directly, and those are called as the direct pathways, or formation of oxidative. I'm trying to see. It's not going to be here. It actually is in the Krebs cycle. This is the stage where you have direct ATP forming, and there are only 2 of those. That's why we have that at the end as the substrate level phosphorylation. So there are only 2 of these. So a lot of the information that we did in this chapter should be a summary of what you've already done in many of these reactions before. Make sure you understand aerobic respiration in prokaryotes. Again, I'm going to summarize it to you that what's happening in here is that glucose is breaking down completely into carbon dioxide and water, and ATP is generated. The process has 3 main phases: glycolysis, the Krebs cycle, and electron transport chain. The preparatory cycle is the one between glycolysis and Krebs cycle, and that's the cycle where you convert your pyruvic acid to acetyl coenzyme A. The key event in aerobic respiration is that electrons are picked from intermediates of of glycolysis and Krebs cycles by molecules that are NAD and FAD, and these are then converted to NADH and FADH2, and these are taken to the tricarboxylic acid cycle, and in where the conversion where actually most of the ATP is generated. So aerobic and respiration, ATP is generated in the tricarboxylic in the electron transport chain cycle, and sometimes it's also called as the chemioosmosis, because it's the movement of electrons from across the membrane. That's why. And another name for that is also oxidative phosphorylation. So you need to know all these terms, because you're depending on what you're reading. Sometimes it will be referring to oxidative phosphorylation, sometimes it's chemiosmotic mechanism, sometimes it will be referred as electron transport chain. Need to make sure you understand that. Make sure you understand lactic acid fermentation, which only takes place in an anaerobic condition. So depending on whether we have lactic acid or alcohol fermentation, they're both anaerobic processes. This concludes our section with microbial metabolism. And aren't you happy that you had done this earlier in other biology classes? So it's not as difficult to understand when you did it again the second time.